not blaming you guys. I think we just need to be mindful of the time and stuff. So.
Just start setting it up. We were talking about setting it up before, like.
Yeah.
Okay.
Okay, folks, we're going to get going. So we're going to switch gears just a little bit and start talking about things related to depression. So I'm going to actually go back and start talking to you again about depression. You're going to have to do a little bit more writing today than you have in the past. So let me take you through a couple of things on the slides and then get you to engage in some of the material as well. So one of the biggest issues right now is that we don't have a good marker for depression. And by the way, I neglected to mention that this is the first lecture that you're responsible for on the final exam. The final exam is not cumulative, so it only goes from this lecture onwards to the very end. So nothing before this lecture, only this lecture and on. So when we looked at some of the different aspects of depression, we talked about some of the different sequelae of symptoms that people developed around depression. And we sort of left things off with some animal models of depression, and then we started talking about different neurotransmitters and how the neurotransmitter hypothesis of depression sort of dominates right now. But most of you will also know that the neurotransmitter hypothesis, that somehow the neurotransmitters in your brain get depressed, and that's one of the reasons why you feel depression related to the change in neurotransmitters. And this goes back to the monoamine hypothesis that we talked about in the last lecture as well. And we talked about how if you induce changes in neurotransmitters by giving them pharmacological agents, reduce the levels by toxicity of certain neurotransmitters, that many of the features of depression occur in these animal models. But there's some really big gaps in that story. So we know, for example, if I were depressed, and I were to get a PET scan and you were to look at my dopamine levels, indeed, my dopamine levels in my brain right now, if I were depressed, would actually be really, really low. The problem with this whole hypothesis is that if I were to take some sort of selective serotonin reuptake inhibitor or selective serotonin and noradrenaline reuptake inhibitor, within about 2 hours in my brain, my neurotransmitter levels would actually be elevated, but I don't feel any better. I still feel depressed. I actually have to keep taking that drug for about 3 weeks to about a month before I get any benefit from that drug. So it's not like you take a Tylenol if you have a headache and a few hours later you feel better. With depression, it takes time for these different drugs. Even though the neurotransmitters go up almost immediately within a few hours, you don't feel better. And that's always bothered clinicians, and it's always bothered scientists for a long period of time. And so people started to wonder in the late 1980s and the early 1990s, well, if we know that the brain chemistry changes with these different drugs, these antidepressant medications, why don't we see these changes right away? Why do we have to wait 3 weeks before we start to see these changes? And so scientists started to think about other molecules that would have to be involved or other pathways that would have to be involved related to depression. A couple of other things, because some of you have already mentioned that you've started your visual summary. Some of you who are doing papers on depression will also know that one of the biggest things that affects students, affect you perhaps, is that cognitively you suffer when you have depression. It's not the mood that really hits students. It's really the depression affects your cognition. You can't sit there and concentrate on what I'm telling you. That's the biggest problem with depression. And one of the things that we know is important is that one of the key circuits, so hopefully in your second year courses, you've learned that within the brain, there are 7 to 8 defined circuits. One of those key circuits actually involves the hippocampus. It's one of those key nodes of this neural circuit hypothesis. And we know that in depression, the hippocampus is actually affected to a really large degree. And we know that we have not only changes in the cortex, so smaller gray matter, thinner white matter, and I mentioned all of that in the previous lecture, but we know that the hippocampus is particularly affected, and your cognition, your memory is affected. The ability for you to recall is affected, especially with depression. We don't know, for example, if this is due to cell loss or if the individual neurons are shrinking, or if it's a combination of that. Are we losing neurons, or the neurons that are in the hippocampus getting any smaller? And this is sort of the background to what we're going to be talking about today. People weren't satisfied with the neurotransmitter hypothesis, so they had to come up with a different mechanism. This is not popular because these antidepressant medications work for about 50 to 60% of the population, so it suggests that antidepressant medications work, but it can't be due to an elevation in neurotransmitters. So let me take you to a slightly different tangent. Let me tell you a little bit about some of the evidence that suggests that it's not only neurotransmitters. Neurotransmitters are important, but they're not the driving force behind things like depression. If you're looking at these two different mice here on the left, the one on the left is actually very different than the one on the right. And you'll see that there is this sort of patch of hair on the top that has changed color. And this is because the hair follicles have actually died here in this region. And the reason that it's died is that the mouse head has been placed under lead shielding. So there's lead shielding here, and there's lead shielding here. And this little gap that you see is where the mouse brain, directly underneath, has been subjected to really high intensity X-ray radiation. And by doing so, not only have you damaged the hair follicles, which is why you get that white band, but you've also irradiated at very high doses this area that we're interested in, which is the hippocampus. And this hippocampus is actually really important. This hippocampus is actually important because it's an area, or one area of the hippocampus known as the dentate gyrus. And this represents the dentate gyrus. And this structure in the dentate gyrus, one region of the dentate gyrus, which is part of the hippocampus, is known as the subgranular zone. And people in the 1980s started to realize that the hippocampus and other areas of the brain were actually involved in what is now known as neurogenesis. This is an area of the brain that even into adulthood actually produces new cells. They have increased numbers of progenitor cells, which are being born every day, which will eventually lead to new neurons. And this is an area that is of some importance. These neurons within the dentate gyrus, as they migrate from the subgranular zone layer, so this, I'm going to use this abbreviation, SGZ, as they start to proliferate and then divide, they will start to also differentiate into full-blown neurons, and they will eventually integrate themselves into the hippocampal circuit. So here's a hippocampal circuit. We know the hippocampal circuit is affected in depression for a variety of reasons. One of those reasons is that in the hippocampus, we actually have one of the highest concentration of glucocorticoid receptors. We have lots of glucocorticoid receptors here, and the neurons respond to stress in this area. So let me also put up this slide here. So I want you to just get some general features of what I'm going to be talking to you about. So this is the hippocampus, the entire hippocampus. So if we're to take a look, we can subdivide the hippocampus into anatomical regions like the CA1 region of the hippocampus, the CA3 region of the hippocampus, and over here, the region that I've been talking to you about, which is known as the dentate gyrus, abbreviated for you here as DG. One particular region of this is known as the subgranular zone layer, and this is where we would find those neuronal progenitor cells. So from the time that these progenitor cells start to divide and eventually start to migrate and eventually form synaptic connections throughout the hippocampus, if you were to time it from today, we would actually find that on average it takes about 3 to 4 weeks. So why is that number important? So why is this neuronal hippocampal neurogenesis, why is all of that important? Because it fits the timeline for what we think is happening in terms of antidepressant medications. They don't work right away. They take about somewhere between 3 to 4 weeks for you to get any beneficial effect before your cognition comes back, before other aspects of not being depressed come back. And so when scientists were first discovering this mechanism, and they were making this correlation that, yes, in depression, the hippocampus in animal models is certainly getting smaller. They have less memory. They have less overall cognitive facilities. It fits in very nicely with what's going on here. And the next few slides are where I'm going to take you through some of the evidence to suggest that this is a mechanism, potentially, for how depression makes the hippocampus smaller, why it affects your cognition, and perhaps some other aspects related to depression as well. For the time being, I'm going to not talk to you about all of this right now. I'm going to mention these in other slides, so you don't really have to go over all of the different points that you see here now. You're going to find that I'm going to cover all of these again in some slides that are coming up. Do you have any questions on this before I go on? Okay, so now I'm going to try to convince you that perhaps this is the reason why it takes so long for antidepressant medications to work, especially in improving your cognition and making your hippocampus not shrink, but perhaps actually survive better. Again, it wasn't formally found until about 1997, and this is Rusty Gage, or Fred Gage, down at UCSD, UC San Diego, who actually found that in the adult brain, not in the developing brain where neurogenesis had been known to happen for a long time, in the adult brain, your brains right now, neurogenesis is always occurring. How many of you believe that right now in adult brains you are capable of producing progenitor cells and therefore produce new neurons? So most of you believe that that's true. But when Rusty Gage actually came up with this theory back in the 1990s, he was actually scoffed at, and no one believed him for a variety of reasons. Can you think of one good reason why no one would believe that your brain could actually have new neurons being formed into adulthood? Yes?
That was kind of the ongoing dogma at the time, was that you didn't have neurogenesis.
Sure, but why the dogma? Absolutely correct. Why not have it? Like, it makes perfect sense now that we understand it, but why not? Yes. No observable structural change? Okay, possibly. What else? Yes. So no good recovery from brain damage, absolutely. And another really key aspect, your brain is actually enclosed within the skull. And where's the room for it to actually expand and grow out? And people were thinking, well, you know, if you had proliferation of cells and you had new growth of cells, wouldn't your brain continually be expanding? We don't see that. And so this idea that neurogenesis in the adult was capable was actually not a theory that was well subscribed to until much, much later. Interestingly, if you go into the literature, especially with drugs that we've talked about before, so drugs that would cause selective serotonin reuptake inhibitors to be blocked. So you increase serotonin outside of the synapse by blocking its reuptake. Things like fluoxetine, also known as Prozac, were shown in the 1990s to actually enhance neurogenesis. So this was actually really cool. Neurogenesis goes down with depression, but if you give an antidepressant medication, it actually goes back up. And so it tries to catch up to what has previously been lost. And so this, again, takes some time. It takes about 2 to 4 weeks for this whole cycle to eventually catch up. So a couple of other things just to make note of, and I've mentioned this to you before. So if we are looking within the hippocampus, and again, this is a different region, but if we're looking at the hippocampus as a whole, and we're looking here at the CA3 cells in the hippocampus, we know that this area of the brain actually has a very high density of glucocorticoid receptors. And so when you are stressed out, these glucocorticoids increase. And as the glucocorticoids increase and interact with the receptors, one of the things that we know happens is that overall the level of a factor known as brain-derived neurotrophic factor, or BDNF, actually starts to decrease. Your brains need BDNF. So every time you learn something new, every time you have something that you find stimulating or that you enjoy, there's a little puff of BDNF that also gets released. And BDNF is something that allows for neurons to survive, to stay healthy. And without BDNF, one of the things that happens is that neurons actually start to shrink. They atrophy, and this has been well described in the literature for a long period of time. So we know that antidepressant therapy or antidepressant medications will actually increase the level of BDNF. We know that it will also increase monoamines, so norepinephrine as well as serotonin or 5-HT. The neurotransmission also goes up, as I mentioned to you before, but that neurotransmitter level by itself is not enough. We also need BDNF, and we need BDNF to be acting on these cells to cause both increased survival as well as growth and more neurogenesis. So again, a lot of studies have also been implicating, using what is known as a meta-analysis, that BDNF in the blood of individuals who have depression and aren't treated by antidepressant medications have also been decreased. So we know that there is a lot to suggest that all of these are linked together. And we know that exercise can actually reverse some of these effects as well. So when we say antidepressant therapy, one of the things that's used now is not just medication, but also exercise whenever possible. So again, this is a really busy slide, and I'm going to take you through what I'd like you to make sure that you cover on this slide. So when we're looking at the neurotransmitter systems, we know that in the normal state, when we're looking at either the release of norepinephrine or serotonin or other types of neurotransmitters, that this actually acts at the level of the neuron itself. And one of the things that happens is as it works on the neuron, that it will also cause the neuron to release BDNF. So neurons need BDNF, and they get BDNF, but they also produce their own BDNF. So this is sort of a positive feedback cycle. The more BDNF that there is, it will actually cause more BDNF by that same neuron to be produced. We know that in a depressed state, for example, one of the things that happens is we lose synapses, we start to have atrophy occurring, and we don't really have quite the level of these different neurotransmitters, and these neurotransmitters therefore don't actually cause the signaling for the neuron to produce BDNF. So again, it's sort of a way to regulate the levels of BDNF. So all of this suggests that BDNF is vital to not just normal healthy neurons, and I'm going to get to neurogenesis in a moment, but it's a really important part of creating a nice healthy brain. In depression, all of this is working against you. So you don't have enough BDNF. Without BDNF, these neurons start to atrophy. There's less neurotransmitters around, so it becomes a vicious cycle that way. So why is BDNF so important? Why do neurons need BDNF? And it turns out BDNF and many of these different neurotrophins actually have a very fundamental role in creating a good environment for neurons to be able to survive. So these are various various neurotrophins, and one of these members of the neurotrophin family is known as BDNF, or brain-derived neurotrophic factor. The original molecule was known as NGF, or nerve growth factor, and subsequently NT3 and NT4/5 have been cloned that were very similar to the structure of BDNF. But I want you to concentrate on BDNF as the factor that we're looking at. So neurons need BDNF, neurons produce BDNF, and neurons produce BDNF to cause the activation and further release of more BDNF. And I'll get to that in a few seconds. So I've just talked to you about a whole bunch of things. Do you have any questions on any of the things that I've just covered before I go on to talk about this signaling pathway? Any questions? No? So one of the key things is if a neuron were to encounter BDNF, which it really needs to have as much BDNF as possible, one of the things that would happen is BDNF will cause dimerization of the receptor. So these receptors typically involve the tracks. So the receptors are either track A, usually for NGF, track B, usually for BDNF, or track C, usually for NT3. So if BDNF were to bind to a track B receptor, the two track B receptors would actually dimerize, they would come together, and when they dimerize, they actually cause phosphorylation. And this is the phosphorylation that you see here. This is actually a key thing, a key step. So the dimerization is one aspect, And after they dimerize, the intracellular regions actually begin to be phosphorylated. Do you know which amino acids, for those of you who are in Biochem 210, which amino acids actually have the potential to be phosphorylated? So tyrosine is one of them, and this is what TrkB receptors do. They get phosphorylated on tyrosine residues. What other residue? Serine is another one, and the last one? Threonine, right? So you have tyrosine, serine, or threonine. In the nervous system, those are very important amino acid residues because all of them have the potential to be phosphorylated. And why is phosphorylation important? Because this is the beginning of the signal that actually recruits different signaling complexes. So BDNF, after binding to its receptor, causing phosphorylation on tyrosine residues, it's only then that all of these different signaling platforms actually become recruited to one area on the plasma membrane, which is what you're seeing here. The most important ones for me that I'd like you to make sure that you are aware of is that after TrkB receptors become activated, one of the pathways that gets turned on is this PI3 kinase pathway. And PI3 kinase, in turn, will activate AKT. Why do you think this pathway is important in neurons? Yes, Andy? Sorry, it activates protein synthesis. Potentially it can activate protein synthesis. What else? More importantly, anything else that you can see on this diagram? So it inhibits apoptosis. Your neurons are actually really weird. Your neurons are actually ready to die at an instant. Like if they don't have BDNF all the time, they're actually programmed to start dying. And so one of the things that's important about BDNF, it actually inhibits this apoptotic pathway here that you see in red. And this apoptotic pathway is where we start to see the importance. Not having BDNF causes these neurons to start wanting to die, and you might therefore start to see the loss of hippocampal cells. So I don't need you to memorize all of the different parts of the recruited complex up here at the top. I need you to know that the AKT, PI3 kinase through AKT, inhibits the internal apoptotic pathway. The other pathways that get activated include the MAP kinase pathway, or the mitogen activated protein kinase pathway that you see over here. And this pathway eventually will cause different genes to become turned on and to become activated. And again, I know that you guys love learning cellular pathways, right? Because you guys all remember those from your first and second year biology courses, right? You do? Okay, so shall we go ahead and memorize this diagram then? No? I'm not a big fan of memorizing this because, again, when I ask my colleagues to write out the entire pathway from TrkB binding all the way down to transcription factor regulation, most of them can't do it. So they'll look it up online. So I don't think that it would be fair to ask you to do it. However, I would like you to make sure that you know that this MAP kinase pathway will turn on a number of different nuclear targets. And the most important one for us is this target known as CREB and this other target known as CFOS. And I'll talk about CREB today, and down the road I'll be talking to you about this FOS or CFOS mechanism a little bit later on. The last pathway that gets activated, this one here, would be to activate the phospholipase C pathway. And when phospholipase C becomes activated, the overall response of the neuron is to increase the intracellular levels of calcium, and this can also have a number of other targets that can all lead to the same thing. So why are all these different pathways important? One, we're turning off apoptosis. The cells are no longer being programmed to die. And in addition, when we turn on both the MAP kinase pathway and we turn on the calcium signaling pathway through phospholipase C, we are activating neuritogenesis. The neurites become bigger. We are also causing changes in terms of the differentiation of neurons. So we start speeding up neuronal cell progenitors becoming pushed toward the neuronal pathway, etc. So I'd like you to make sure that you spend some time on this slide and that you know the different aspects of neurotrophin signaling. So a couple of other things. I've mentioned to you this pathway here related to BDNF, and BDNF is reduced in stress. Another factor that is reduced by stress in your brain is VEGF. Now, why are these two factors so important? It turns out that this other factor, which is known as VEGF, or vascular endothelial growth factor, actually increases proliferation. Stress is actually lowering VEGF, which means that overall the progenitor cells can't proliferate. They don't have the factor to turn them on. So again, suggesting that that hippocampal pathway, that dentate gyrus, is really important. So we need VEGF to increase proliferation within that subgranular zone layer, and we know that without BDNF, we actually don't have that cell survival. So we need both. And stress, what it's doing is actually lowering the levels of both VEGF as well as BDNF. And these, again, are really important factors. We know that antidepressants are actually much more complicated than what I've just indicated to you. The simplistic version is that somehow, through monoamines, through neurotransmitters, they work to increase the production of BDNF. But we know that other factors are also activated, not just BDNF. Antidepressants will also increase VEGF. Antidepressants will also increase this other factor, which is known as insulin-like growth factor 1, or IGF-1, as well as basic fibroblast growth factor, or FGF-2. All of these, when they're upregulated, will lead to increased neuronal function, better synapses, more spines, more neurogenesis. And we know that the opposite is true. When stress dominates, we have less neurogenesis. We don't have as many spines. We don't have as many neurites as we normally would. So all of this suggests that these are really important overall mechanisms. Is there anything that I can clear up or explain to you more now before I get on to the next slide, or anything that you want to share or discuss? Yep.
What did you say neuritogenesis was?
Do you remember those microglia that we talked about, and they have branches? So neurites are like the branches on neurons. So having more neurites means that they have more branches. So overall, when you have more branches, it means that you likely have more synapses. Nope, you can use those terms interchangeably, arborization or neuritogenesis. Neuritogenesis meaning you have new branch growth. Any other questions or things I can clear up for you now? Okay, so here's a really busy slide, and I'm going to take you through this really busy slide, and there are specific aspects of this that I want you to make sure that you know. Here we're going back to the dentate gyrus, and when we're looking at the dentate gyrus, we know that there is a population of cells that will undergo neurogenesis in what is known as the subgranular zone layer, the SGZ that I talked to you about before. Within this region, and only in this region, because it's different, this is a very unique form of neurogenesis. One of the things that we have are these progenitor cells, which look an awful lot like the developmental form, like when your brain is expanding during development. As you go from an embryonic to a fully developed fetus, your brain expands dramatically. One of the structures that's really important in that developmental growth are these radial glial cells. And within the subgranular zone layer, in the adult, these are still there. And these radial-like glial cells are believed to be the progenitor cells. So these will eventually form these intermediate progenitor cells that you see here, the IPCs. So these radial-like glial cells will convert themselves into these progenitor cells. And if they are pushed along by the appropriate signals, these progenitor cells will also then form neuroblasts. The neuroblasts are the first stage in forming these fully differentiated neurons. They're already looking like they're going to form these neurons. And one of the reasons why we know that this is true and why it's really important, early on in development, as you likely remember from your second year neuroscience courses, the very first neurotransmitter system to come online is GABA. So there isn't a lot of excitation initially. And GABA is the first sort of system that comes online. And these neuroblast cells actually have GABA A receptors. So we know that they look a lot like early developmental cells. So these GABA A receptors in yellow become really important. Now, early in development, as you likely remember, one of the key factors that make GABA excitatory is the fact that we have a difference in the levels of ions inside the cell versus outside of the cell. And the reason why they're different inside and outside of the cell are due to all of these different, differently expressed co-transporters. And these co-transporters that you see here are NKCC1 as well as KCC2. Now I have an abbreviation here at the top. These are all co-transporters. Based on what you remember, I hope you remember, from either one of your second year courses in biology or one of your neuroscience classes, do you remember what ions are getting co-transported? And you could probably take a really good guess based on this NKCC abbreviation. Sodium, potassium, and chloride. So we have three different ions being co-transported. And in addition to that, we also have KCC2. Does anyone want to make a guess as to what gets co-transported with KCC2? Sorry? Potassium and chloride. So all of these things are being co-transported. It looks an awful like early development, and these neuroblasts have all of these different things that look like an early developmental neuron. So it has GABA A receptors, it has a whole bunch of these co-transporters, and eventually, over the course of time, so remember this whole process from start to finish takes about 3 to 4 weeks. It will continue to differentiate. It will start to make connections with these inhibitory interneurons that are always there. These are always present. They're not just coming up. And these will start to make these different connections. Eventually, they will form what is known as an immature dentate granule cell. And this dentate granule cell will start to make synaptic connections with all of these other synapses that you see up here at the top. So this is an axon, and this axon is coming in, and it's making one synaptic contact here, and it's making another synaptic contact there. Now, do you remember what sort of neurotransmitters get released the most often within the hippocampus? Thank you for telling me. Glutamate, yes, awesome job. So glutamate gets released, and this glutamate that gets released interacts with these NMDA receptors that we start to see being expressed. Again, this whole process, which is really what I want to get across, so I'd like you to make sure that you know the different molecules that are found on neuroblasts and that are found on these different stages are really important. The NMDA receptors, not so important right now. We'll come back to that later on. This whole process takes about 3 to 4 weeks, which is why it's really important for us. All of this needs VEGF, it needs BDNF, it might need IGF-1, it might need FGF2, but for sure it needs BDNF, and for sure it needs things like VEGF. Any questions or things that you want to make sure that you need to know or don't need to know on this slide before I go on? No? Okay. So again, it's a really busy slide. I don't need you to know all of the details on this slide. There are some other things coming up. This is probably one of the most sort of controversial areas. Today's lecture, by the way, is mainly about the controversies that exist in neuroscience. I told you earlier that Fred Gage down at UCSD, I think now he's at the Salk Institute, came up with this idea of adult neurogenesis. Most of you now believe that that's true. But this is actually very contentious, this diagram up here. We know, and there are people here at U of T that actually study this, we know for a fact that these three areas of the brain now have neurogenesis. We know, for example, in the hippocampus, in the dentate gyrus, we have hippocampal neurogenesis going on. There's another area that I'm going to concentrate on, which is known as the subventricular zone. So the cells that line the ventricles of the brain. We also have neurogenesis going on in this level of the brain as well. And this other area, which is known as the hypothalamus, has also been shown to have neurogenesis going on as well. And again, you don't have to worry about all of these details. It's just to let you know about these three areas, which is really what the purpose of this slide is. 3 areas in the brain where we have defined neurogenesis going on in the adult. Any questions on this before I go on? But this is really controversial. And this, again, becomes one of those big issues. I talked to you about this when we talked about the microbiome. Do animal models actually recapitulate? Does it actually reflect the human brain really, really well? So I'm not going to go through this in a lot of detail. The bottom line is that the animal models that we use, the mice and rats, for the most part, over here on the right, actually suggests that they're fairly good models. What we see in the human and in the rat or mouse model is pretty accurate. We see a lot of things being reflected. So for example, we know that here in the neocortex, for example, that for the most part, we don't really see neurogenesis going on here. We don't have constitutive all the time going on in terms of neogenesis. We don't see that in the human brain either. We know that the hypothalamus, we see it. We know the dentate gyrus, we see it both in humans as well as in the animal models. And we know that the subventricular zone, we see it both in humans as well as in animal models. This other area, which is down here at the bottom, again, the striatum is actually one of those areas that is under dispute. In animal models, it turns out that the striatum actually has continual constitutive forms of neurogenesis. People see it in rats, but they don't see it in mice. We know that if you damage the striatum, that we increase neurogenesis. So that's reactive neurogenesis. You damage this area of the brain, neurogenesis increases. However, in the human brain, we actually don't think the striatum actually is a source or a place where we see a lot of neurogenesis going on. We don't see new neurons, whether it's constitutive or whether it is reactive. So there are some slight differences, but for the most part, these animal models tend to be fairly good. So again, just to go back to a couple of key points, overall, when we're looking at neurogenesis, one of the things that we're finding fairly routinely, the ones that are just being born, the ones that you are generating now, the ones that were born about 3 to 4 weeks ago, those are the ones that are actually the most excitable. They're the ones that actually have the most synaptic modifiability. And we know that that's an important way for you to be able to learn. And these, again, are readily activated by new learning experiences. So imagine you've just turned all of that off, right? So you're depressed. You don't have this system, this reservoir system available to you. You don't have these really rapidly modifiable synapses available to you. It becomes harder and more challenging for you to learn something new or to retain something new. And again, we think that these newly born adult neurons are really important for certain forms of learning as well as memory. And in depression, you're losing all of that for a variety of different reasons. Again, we also know that as you get older, and this becomes a really key point in the second part of today's lecture, that as you get older, unfortunately, neurogenesis starts to decline. Why do you think that happens, by the way? Why does neurogenesis get lower and lower as you get older? Any ideas? Yes? Wear and tear. But it's inside your brain. It's a good idea. Wear and tear. Anything else? Yes? Okay, so maybe the repair mechanisms aren't working. There's something going on. Eventually, they just run out of gas or they start building up more errors. Anything else? Yes? Less BDNF as you get older? Potentially. So all great ideas. And I'm going to get into this again a little bit later, but it's almost universally true that neurogenesis as you get older, unfortunately, starts to decline. We know that in Alzheimer's disease, in schizophrenia, and in many other neurodegenerative conditions, neurogenesis is also affected for a variety of reasons. And we know that stress-induced forms of depression also dramatically reduce neurogenesis as well. So people are interested in this for a variety of reasons. There are many labs here at U of T that actually deal with neurogenesis and different aspects of neurogenesis. So I'm going to take you through a couple of really key points in going from these radial glial-like stem cells into the proliferative state, how they survive. Do you remember what factor causes increased proliferation? VEGF, thank you. And what causes increased survival? So the BDNF. So we need both of these to work on the proliferative versus the survival. It really makes a big difference, and I'll show you that in a few moments. And again, this whole time course takes about 3 to 4 weeks to form this mature adult born neuron. So there's a couple of terminology points that I need to make sure that you're all clear on. When we're looking at neurogenesis, there are some key markers. How do we know that this neuron is just newly born versus one that's already been there for a while? Like it was there since the time you were born, it just didn't appear like in the last 5 minutes. How do we make that distinction? One of those things that makes this possible is that we have a marker known as double cortin, and you can see double cortin positive cells here in the light color, and you can see some of the branches of the arborization or the neurites going up here at the top. And double cortin is a marker for neurogenesis. It's only cells that are undergoing neurogenesis that express double cortin. A fully mature neuron actually doesn't have double cortin at detectable levels. These are also true for when neurons are migrating. When neurons migrate, which is during neurogenesis or during early development, we see the expression of double cortin. There's this one other aspect that I need you to be aware of because I'm going to talk to you about this in a few moments. We can also inject in a substance known as BRDU, and BRDU is a thymidine analog. Do you remember in Biochem 210 you learned about DNA synthesis? Kind of remember DNA synthesis? So if I were to inject in a thymidine analog, why would that be important? Like how could I actually use that to my advantage? Yes.
Only newly formed neurons will incorporate it into their DNA.
So how do we get those newly formed neurons? So if you were to pick a spot here, where would the thymidine actually get incorporated into that DNA? At what stage? So when they're dividing and when they're proliferating, they take up this thymidine analog, and we can actually track at this proliferation stage. We inject in BRDU, only the cells that are dividing take up BRDU, and only the cells that are proliferating are dividing within the brain that are picking up BRDU, and then we can go back and track using an antibody, does this cell have BRDU? So we have lots of cells that are migrating, lots of cells that are relatively new, but at the time that we injected BRDU, only this one cell was dividing at the time, which is why we're seeing it in this particular picture. Lots of other cells are still undergoing neurogenesis, but at the time that we injected, only this one cell was able to take it up, and this other cell down here at the bottom, it's the only cell that remains in this mature state. And the calbindin is something I'll talk about a little bit later. You don't have to worry about that right now. Do you have any questions on this before I go on? So we have ways to mark the proliferation of cells using BRDU. We can identify migrating neurons or neurons that have undergone neurogenesis using things like double cortin. Any questions? Just to make sure that you're all clear on this. Do you remember the 3 different regions of the brain that I told you are not disputed for neurogenesis? So dentate gyrus, 1. Hypothalamus is the other, and the subventricular zone. Would I be able to see double cortin staining for neurogenesis in all 3 of those brain regions? What do you think? Yes, so all of those would work there. Would I be able to see BRDU in those dividing cells? Yes, so they are ubiquitous. We can see them everywhere. But I also told you, and then we'll take a break after I go through this one slide, I also told you that there are some unique properties to these other regions. The ones that are really interesting here in Toronto, because it was actually discovered or co-discovered here in Toronto as well as in Calgary by Dr. Moorshead, that there's a region of the subventricular zone. So these are the ventricles, the left ventricle, and the lining of the cells that surround the ventricle. If we were to take a look, there are these subependymal cells that are in this lining, this gray region that you see here, that actually also undergo neurogenesis. And these are really important because they don't look at all like the neurogenesis that occurs within the hippocampus. And there's so much of this research that's been going on here in Toronto. So I want to take you through some of her research in the next few slides. And I think for the time being, we're just going to take a quick break, and then we'll come back and pick up the rest of the story. So I think, Max, I'm going to need your help just a little bit. For today, you don't actually have to stand up. Most of this can actually be done seated. It's more of a mindfulness sort of exercise. Do I need to switch this at all?
Nope. Should be fine. Should be fine. Yep. And where did it go? It'll come. There it
My
is.
name is Michaela, and I'll be taking you through your mindfulness movement break. The intention of this break is to slow down and bring attention and care to the body and to check in. The invitations here are to move only as far as is comfortable and safe, and not to stretch to the limit so much, but really to become aware of the different sensations that are associated with each movement. To begin, allow your eyes to close, or if that's not comfortable, allowing your eyes to be open but softening the gaze. And when you're ready, bringing your chin to the chest, just as far as is comfortable. Noticing any sensations that are associated with this movement in the back of the neck, shoulders, upper back, mid back, low back. Just taking stock. Allowing your head slowly to roll towards the right shoulder. Noticing any new sensations that arose as a result of this movement. Allow your head to fall backwards. Noticing a new location of sensations. Upper neck, lower neck, shoulders. Allowing the head to fall gently towards the left. And whenever you're ready, allowing the chin to come back to the chest. Continuing at your own pace in the same direction. The intention here is not to evaluate or wish that the sensations were different, simply just to notice what's there without needing to do anything about it or change it in any way. When you're ready, allowing the head to roll in the opposite direction. If you notice that your attention wanders away from the sensations in the body, simply acknowledging where the attention went. Perhaps to thoughts of the future, thoughts of the past, perhaps to thoughts about this experience in this moment. Just gently acknowledging where the attention went and bringing it back to noticing the sensations in this moment. When you're ready, bringing the head back to center and taking a moment here to notice any after effects of this stretch.
Okay, awesome. So we'll take a quick break and then we'll come back in about 5 minutes. Thank you. You guys normally just keep that right?
Sure, yeah. It's working, but yeah.
Okay.
Well, I don't have some figures for actual experience timeline. I
We'll get that. You have to cite where your figure sources are.
yeah, we take all these figures from just your paper at home. We're just doing one large citation. We just do individual.
Probably just say all figures taken.
The timelines are alright.
Not sure. So it's too really busy with these slides.
Should I simplify it? Isn't that like this?
What do I really need to know from that one graph? What's the key point there?
It shows that it's diffuse integration.
Okay, so why do I need to see all of this?
Because the results from it show three different results. You can highlight every single image.
You don't have to highlight it.
You pick the ones that you think that's the most important one.
Correct. Okay. Hello. unfortunately, VEGF, but BDNF doesn't seem to.
And also, do you think that when in the striatum they have neurogenesis? So, if I'm remembering correctly, in the striatum, what if your tendon just creates like a fat block on a function if they have neurogenesis?
I think that'd be really interesting. I don't know of any off the top of my head, so that'd actually be a really interesting question.
Yeah. But like for most of these functions, they're the same. So when you look at like whether they have neurogenesis diseases,
Yep.
is it like a post-mortem or
Nope.
how do you do that? Like how do you take a like what?
Oh, how do you know that? So, how do you know that neurogenesis is occurring?
Yeah.
So there's this really interesting, and it is post-mortem, but even post-mortem, how would you know?
Yeah.
Okay, so there's this really interesting case from a while back. Do you remember in Russia there was an explosion at the Chernobyl
Oh, the
nuclear plant?
radiation.
That radiation actually got into the brains of these individuals that were living around. Postmortem analysis actually showed that the regions that we just identified also had radioactivity there in those specific areas.
Oh,
And the only way that they could have it in those specific areas is if neurogenesis had occurred. Because it's not like they get continual exposure to that
yeah.
radiation. It was there, they ran,
Yeah.
and
And
then
it was still there. Oh, okay,
yeah.
okay.
So it does have a sort of mirror
Yeah.
to the human condition.
Can you maybe repeat your question? I just kind of skipped over it. Ryan.
Ryan?
Ryan.
Ryan. Can you resend that email?
No, 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 no. You acknowledged it.
Okay.
It's I meant to send it, and I think I made a mistake.
Yeah, just send me a thing again. I've had a lot of emails over the last couple of days, so I'll go back and check tomorrow morning if I
You acknowledged it slightly.
I probably haven't followed up on it, so I might have acknowledged it at the time, but Brian, it sort of skipped my mind.
I want to talk about my
Yep, you probably want to do that in the next couple of days. Just send me a quick email along with your discussion notes.
I have one question. They're saying that would this be okay as a human animal to nature nature but clinical studies or
So the clinical study is okay.
okay. So even though it would be like a neuroscience article?
Yeah, it's fine. As long as it's a primary paper that's been published in the last 5 years, I don't really care.
Okay.
It doesn't have to be from Journal of Neuroscience. Okay, if you guys are okay, we're going to get going. So I'm going to take you through some different aspects of what the stem cells that are part of the subventricular zone, what they actually do. They actually form a very specific group of neurons, and these will eventually migrate to the olfactory zone region, and they will eventually become interneurons within the olfactory bulb. Same thing in humans, as I mentioned to you before, and many of these things are actually very, very similar. So we know that within the subventricular zone region here, that in the entire length at the level of the subependymal zone, there are specific groups of cells which will eventually form the neuronal lineage. They form new neurons, in other words. And when they start to proliferate and then they start to migrate, and again, when they're migrating, these will express double cortin, etc. As they go from the subventricular zone, they will actually migrate on a very specific path known as the rostral migratory stream. Some of you who go on to take CSB 430 neurogenesis will actually talk about this with Dr. Tropepi, and he'll talk to you about this in much more detail about all of the different interacting molecules that are found in this pathway. It's a lot like walking from here across the grassy area in front of UC and in Con Hall. You'll see that there are little pathways that people have created, and it's the same idea that there is a rostral migratory pathway or stream that all of these neurons, these newly born neurons, take as they reach the olfactory bulb. It's thought that this is really important, especially in female rodents, because they need to be able to identify and smell their pups, the newborns that they have, and they increase the number of neurons here, especially right after birth. So they have to be able to detect their own pups, etc. So again, I'm not going to go through all of these different steps, nor do I want you to know all of these different identified markers. I don't think that that's what the scope of this particular slide entails. But one of the things that I wanted to introduce to you in terms of a key difference, here we're looking at an astrocyte as the putative stem cell. So it's a little different than the radioglia. In some ways it's similar to, but a little bit distinct. Here we're looking at a subventricular zone astrocyte, which becomes this putative stem cell. The only marker that I'd like you to make sure that you are aware of, because we'll see this later on, is this marker known as nestin. So nestin is a protein that is specific for these progenitor cells that are found within the subventricular zone. It is also a general marker for stem cells in general. So nestin is that key protein or that key marker that I'd like you to be aware of. These other ones, for the time being, we're not going to discuss, although you'll see things like double cortin that you are probably remembering from a few slides back. So let me tell you a little bit more about these stem cells. So if I were to take a single cell, and this is what Dr. Morsehead had done initially, if I were to take a single cell from the subventricular zone and I were to scoop out the subependymal layers, so you can actually strip them off, peel them off, and if I were to get them down to a single cell, I would actually be able to see that they divide. And if I were to leave them in a cell culture dish, they don't become flat. They actually form these little spheres that actually float. And the single cell will actually form this sphere in the presence of media which is known as EFH. So this is epidermal growth factor, or EGF. It also contains FGF, or fibroblast growth factor, as well as heparin. So if I were to do that, I would get this overall neurosphere that would be floating in culture. And the properties of this being a stem cell, I can take the entire neurosphere that I have here, and if I were to break them apart in EFH, again, one of the things that would happen is I would have individual cells, and if I took any one of these cells again, they would also be able to proliferate. They would also form their own neurosphere. So they're capable of that self-renewal and that proliferation, which is a key aspect of what a stem cell is. And again, for the sake of our discussion, we'll just call any stem cell a progenitor cell or a neural precursor cell. Again, I'm not going to get into the fine definitions of this. The thing that makes this really unique is that each of those cells that are in this neurosphere actually has the capability of building all of the different cells that you have in your brain. And that's why we're interested in neurogenesis overall. So we know neurogenesis is affected in depression. We know that there are different factors that can actually go in and change neurogenesis, but we're interested in some of the basic mechanisms of this because in theory, we can actually take these different neurogenetic precursor cells and actually form an entire brain. So every single cell that we would have in our brain, including having astrocytes, which are marked by the protein GFAP. So GFAP is a marker for astrocytes. We can also, if we differentiate them with a specific media known as fetal bovine serum or FBS, they will also, each of these will also form neurons in green, and we can detect mature neurons by a marker known as beta-3 tubulin. And we can also detect using the O4 marker oligodendrocytes. All of the different brain structures, brain cells that you have exist as these progenitor and these stem cells. And that's one of the reasons why we're interested in it. All the things that we've looked at eventually lead to neurons, but that doesn't mean that they don't have the capacity to form other cell types. Do you have any questions on this before I get on to the next slide, or anything that I need to go back and explain a little bit more? Yes, Zoe. Fetal bovine serum. Have you grown any cultures? Cell culture labs? No cell culture labs? Cells don't actually grow unless they have proteins. And fetal bovine serum is something that you take from a young cow, for example, a calf, and you can actually take their serum out, the blood portion, the liquid portion of their blood, and in the liquid portion of the blood, we have unidentified growth factors, which we don't know. We don't know all of them. And this fetal bovine serum causes these stem cells to differentiate into neurons, oligodendrocytes, and glia. So I would strongly recommend learning how to do cell culture because it's one of those things. You can't just grow them in media. You need actually serum in your media as well. So I'm going to switch gears just a little bit. So we started off talking about depression and its overall effect on neurogenesis. Neurogenesis goes down. We talked about the basic mechanisms of neurogenesis and why neurogenesis is important. And I mentioned in passing that one of the things that works really well, in addition to antidepressant therapies or medications, is also exercise. And this goes back to what we know. And we've done a lot of research here at U of T as well. So there are people in physiology like Dr. Wojtowicz who actually look at these different processes. If we were to compare on the left series of panels versus the right series of panels, a normal mouse versus a mouse that we see here who is forced to do some exercise, then one of the things that we can see is that this mouse actually has neurogenesis. This is the dentate gyrus. We have injected these mice with BRDU, so only the proliferating and dividing cells. So each of these little black spots that you see here actually show that cells have started to divide and therefore proliferate. An exercising animal actually has much more proliferation. You can see that very, very clearly. 12 days after BRDU injection and following exercise, we see many, many more of these spots in the dentate gyrus. So proliferation goes up for sure. Do they survive? Because remember, proliferation is only one aspect. Proliferation is run by VEGF versus survival, which is run by BDNF. So if we continue many, many weeks later, of those that were originally proliferating, how much of those that started to proliferate early on are actually left? Very, very few. So you start off with thousands to tens of thousands of these neurons at any given point in time, but at the end of, for example, 5 weeks, we only have maybe about 2 or 3. It's not really beneficial to us. But if we were looking at the effect of exercise, so not only did we increase proliferation initially, but we've also increased survival dramatically. So instead of having like 1 or 2 of these neurons, we have many, many more of those neurons still surviving. And remember, those are the neurons that are more easily modifiable. They have better synapses. They have synapses that can learn better. And we think that these are really key and important types of new neurons that are being born. So exercise actually leads to both proliferation increases as well as survival of these different stem cells as part of neurogenesis. We know that if I were to ask all of you, and I'm afraid to ask all of you because I'm not sure you'd answer me, but if I were to ask all of you, do you think exercise is good? I would imagine that most of you would say yes, exercise is great. Everyone should be doing and engaging in exercise. And we know that regular exercise actually has many, many beneficial effects. So it will reduce cardiovascular disease risk. It will change metabolic disease risk. We know that it has a lot of anti-inflammatory effects that I'm going to get to in a few moments. And we also know that it will change your visceral fat mass, the fat that you have just under your skin at the level of your belly, for example. So this also means that you are going to be changing what is released from those fat cells. You reduce the fat cells, the number or the size of the fat cells. You change the release of factors from those fat cells, which are known as adipokines. And this means that you are producing, with exercise, different adipokines or reduced adipokines, and you're also increasing anti-inflammatory factors that are getting released as well. So exercise has some really powerful benefits. And we know that these anti-inflammatory effects in exercise are actually really important to us. In fact, you can over-exercise. And it turns out that if, for example, you're a varsity athlete or an Olympic athlete, if you over-exercise, you can actually take the anti-inflammatory effects that are being produced through exercise to a level where you're actually producing a state of partial immunosuppression. So you might have this really, really healthy young individual who's part of a varsity track team or some other sports-related team, and they get sick the most often. They're probably the most healthy person, but because they've actually induced anti-inflammatory processes, they actually might be more susceptible to different types of infections, especially virally mediated infections. So again, physiologically, exercise might have a lot of anti-inflammatory effects, and we know that many chronic disorders like type 2 diabetes, cardiovascular disease actually are mediated through inflammation. So having anti-inflammation to treat type 2 diabetes or cardiovascular disease makes a lot of sense. But it's also been shown for quite a long time now that exercise has a number of different beneficial effects on the brain. So both in terms of mental health as well as on potentially like long-term effects like depression, as well as Alzheimer's disease has been implicated, etc. But for the most part, it was almost like being in the realm of psychology. So in psychology, people ascribe a lot of different phenomenology, but the mechanisms take a little bit of time to tease apart. Neuroscience wants to understand the mechanisms first and then try to figure out what the overlying or underlying behavior is. And until 2013, no one could actually link why exercise was actually beneficial to your brain. All these other things, the effects on type 2 diabetes, reducing inflammation, reducing cardiovascular disease, all of that made sense. But in terms of why your brain gets better, why you can think better after exercise, until 2013 was actually a big unknown. And in 2013, there were a couple of different events that occurred. Very controversial events, by the way. So I'm going to introduce some of these different factors. So we had known for a long time that with acute exercise, so you go out, you do a sprint or you do exercise for a couple of days, we know that in your blood we have a number of different factors that actually spike. They go up. We know your growth hormone levels go up. Another molecule known as prolactin go up. As you exercise, your muscles get warmed, and so we have elevated levels of heat shock proteins. And we also know that other factors that influence how leukocytes or white blood cells traffic to areas of infection also get affected by exercise as well. But we don't really understand all of the different factors. We don't know which one of these factors is really beneficial to your brain. And so this current model was actually developed around 2012, just before the papers that I'm going to be introducing to you. When you look at exercise, the main effect of exercise, if you're doing it appropriately, so not like me, I exercise by watching other people exercise, but you're actually going out there and your muscles are contracting. As your muscles contract, your muscles are actually an endocrine source. Your muscles actually secrete as they contract. Every time your muscle contracts, it releases compounds like interleukin 15, interleukin 6, IGF-1. Do you remember IGF-1? It also releases VEGF, and these are all known as myokines. These are factors that act a lot like hormones that come from contracting muscle. There's this other factor here at the top known as irisin, and I'm going to talk to you about irisin in a few moments. This can also directly affect the adipocytes that you see over here. Now, you don't have this on your slide, so you might want to jot this down. You don't have interleukin 10. So muscles and myokines can actually reshape some of the different adipocytes, which is what you are seeing here. Adipocytes are fat cells, particularly visceral fat, the one that is around your belly, will actually have release adipokines that I mentioned to you earlier. Much like myokines from muscle, your fat cells are always secreting factors as well, and factors that can work to your benefit, including like adiponectin. Adiponectin is one of those factors that seem to be changing as you change your obesity levels, etc. And they will also produce other factors like interleukin 10 as the adipocytes start to shrink. So as they start to shrink, they actually reduce or produce interleukin 10, which is an anti-inflammatory factor. All of these different things coming together have been thought to enhance neurogenesis, thought to enhance synaptic plasticity, do that dendritic remodeling, increase hippocampal plasticity, and as a result, exercise through all of these different pathways has been thought to increase learning and memory, right? And again, somewhat controversial, but really relevant to our discussion today. So this is where things get really dicey. This is where the model actually becomes both interesting and a little bit scary. So I'm going to take you through two of the biggest controversies right now related to myokines and how these myokines, things that are released from muscle, actually might be working to change your brain and why it's so controversial. So I don't know if you've read this a while back, but this actually made really big news. People wanted to know, when you exercise, like, what's the difference? Why do young people actually stay young? Why do people who tend to exercise look different than people that don't exercise? There must be something that gets into the blood. We know things like growth hormones are really important. We know prolactin's important. We know a whole host of factors. Let's try to identify some of those factors and try to identify why this becomes important. And so people did some bizarre experiments. How many of you have heard of parabiosis? Awesome. No one. Okay, it's going to be a fun lecture. So let me tell you a little bit about this study, which is actually based in science, and I'm going to show you this in a few moments. How many of you have heard the study where if you do a blood transfusion of a young individual into an older individual, that the older individual feels better, thinks better, etc.? Some of you have heard that. So this is actually based on a study that came out of Amy Wager's lab, and I'm going to go through that in a few moments. It actually tended to rejuvenate the older mice. The older mice actually looked younger again. It was actually one of those really cool stories. It actually made its way into, for example, popular media. People do this now. So how much would you pay to have a blood transfusion and to feel younger? So it costs about $8,000 in the United States. So they take people who are between the ages of 15 to 25. So if you want to make fast, quick money, go out and donate your blood now. Be nice and healthy. Be prepared to be one of these individuals that gets hooked up and does these transfusions. And these young individuals are in very, very high demand, as long as you're healthy. Okay? So this goes back to, why do people want to do this? Where's the science behind all of these different studies? And this actually comes from this study here. And I'm going to take you through this study. I want you to understand the basic premise of this and why this is incredibly controversial. I know you don't have this on your slides, so you might want to jot this down as well. One of the things that you can do is you can actually very, very easily, it doesn't take a lot to do this, you can make a small incision in the skin into the subcutaneous layer, and you can actually sew together two different animals. Their blood systems, their vasculature will actually grow from one side to the other across the cut, and as a result, their circulatory systems actually start to intermingle. This is what is known as parabiosis. But you can take that one step further. You can create what is known as heterochronic or different aged parabiosis, which is what you are seeing here. You can take an older animal and connect their circulatory system to a younger animal that you're seeing over here. And what happens if you were to examine the younger animal's brain and you were to allow the older animal's blood to mix with the younger animal, is that in the younger animal, it actually functionally looks like an aged animal. So it might look like it's a lot older than it is. There would be decreased neurogenesis in the brain of this mouse over here. There would be impaired synaptic plasticity. It wouldn't remember things as well. It would functionally look like an older animal. If you allowed it to go the other way, and I won't get into all of the technical details, but if you were allowed it to go the other way and take the blood and the circulation to allow whatever factors were making this mouse young to go from the blood into the older animal, you would find that in the brain of the older animal now, that it would look like it's rejuvenated. We would actually see an increase in neurogenesis. Remember I told you neurogenesis declines with age? This gets reversed if you get a transfusion of young blood. We would also see, and you can actually see this here, increased synaptic plasticity, or LTP. So this is the normal level, but now after rejuvenation, we get this much bigger level of synaptic plasticity, and we now know that there's actually a really strong effect on cognition. They remember things better, they learn faster, it's a much better system. So there's a couple of really interesting points about this. And again, if you want to go and check out the actual paper, it was published in different aspects of stem cell, and I can send you other links as well. But I wanted you to think a little bit about this. There has to be a factor that's in the blood, right? So you're switching the circulatory systems from one animal to another. You're not transplanting muscle. You're not doing anything else. You're just allowing a blood-borne factor to go from mouse A to mouse B. That is parabiosis. The factor that was identified as GDF11. And this GDF11, this growth differentiation factor 11, was thought to be the rejuvenation factor. That's the thing in blood. As you get older, your GDF11 levels go down farther and farther. And this is what causes your brain to look older or not to function as well. Sound good? No? Sounds good to me, right? So I'd go out and I try to create as much GDF11 as possible. Again, you don't have to worry about all of this here. It's just for you to kind of go back and take a look at. This shows you exactly the same things that I showed you on other slides. People have done parabiosis for a long time. People know that this is true. The problem is GDF11 true? So the parabiosis, there's absolutely no doubt. If I were to have one of you volunteer to donate your blood to me, as an older individual, I can't pay you $8,000, by the way, but if you were to donate your blood, I would actually be a lot better. I would be a lot smarter. I would be able to have increased neurogenesis. That part is true. Is GDF11 actually true? This becomes the really big source of controversy. As soon as that paper came out, other authors actually came out and said, you know what, this is completely wrong. The results that you've found in this particular paper actually suggest that you are picking up the wrong molecule. That in fact it can't be GDF11, that GDF11 looks a lot like this other molecule known as myostatin, and myostatin actually does the exact opposite to what GDF11 should be doing. So this created this huge controversy. And right now, since that paper came out in 2014-ish, actually, there's been no further work. In fact, you can go to Amy Wager's website. She's at Harvard, and you can actually go in and see what her research is, but there have been no follow-up papers. We don't know what causes or what factor is in blood. It has to be a blood-borne factor that actually rejuvenates the brain It still exists, but no one actually knows what it is. But it's very unlikely that GDF11 is going to be the molecule. I introduced it for you, and you can go and read this yourself to understand the controversy. You're not responsible for it, but this is what neuroscience is. It's all about the controversies and trying to figure out how do we improve the brain and neurogenesis, whether it's for depression, for neurodegenerative conditions, or just plain old aging. How do we increase all of this? And if exercise is that important, it still doesn't answer like why exercise is so crucial to resetting the brain, as many people have also found. Until this story came out. So this is that story that I'm also introducing to you as the second big controversy right now. So remember I told you that the muscle, as it contracts, actually releases these myokines. This all goes back to the fundamental molecule found within muscles. And within muscle, there is a particular protein which is known as FNDC5, or fibronectin domain containing protein 5. This is elevated during muscle. This has been shown by many, many labs to actually be upregulated. This is the overall structure of FNDC5, where you have a signaling molecule and then this fibronectin domain, and then it's embedded within the plasma membrane. It gets proteolytically cleaved, and one of the things that happens is this N-terminal region, we call this N-terminal region irisin. And so it looks an awful lot, because it has a signal peptide, it looks an awful lot like having a pre-pro hormone, just like insulin is produced first as proinsulin, and then it's processed into insulin itself. And this fragment, irisin, as it gets cleaved, and this would be out in the bloodstream, so this is in the membrane of the muscle. This is outside the extracellular region. This gets into the bloodstream, and then irisin might actually work on a number of different tissues once it gets into the blood. So again, this is really important because things like brown adipose tissue actually might be protecting and converting white fat into brown fat that protects against diseases like diabetes as well as obesity. So this is what happens when you exercise. You increase the amount of FNDC5. As you make more and more FNDC5, it's more likely that you're going to produce more and more irisin. As you produce more and more irisin, you're going to be able to convert white fat into this brown adipose tissue. So I'm going to come back to FNDC5 in a few moments, but there's some other research that kind of interplays with this. There's another molecule that plays into this role with FNDC5, which is known as PGC1 alpha. So one of these individuals is Bruce Spiegelman, also at Harvard University, and he was noticing that if he were to knock out this molecule known as PGC1 alpha, and you'll see why PGC1 alpha is important in a few moments, he found that they were really hyperactive. These mice, when you knock this thing out, actually started to become more and more active, were staying up at night, being more aggressive, and also had brain lesions. Their brains were affected by knocking out PGC1 alpha. And one of the reasons why this is important is that both FNDC5 and PGC1 alpha are kind of interlinked. And I'm going to go through that pathway in a few moments. And it's not just that PGC1 alpha and FNDC5 are co-expressed in muscle, these are also expressed within the brain. And so this whole idea was that Bruce Spiegelman developed was, since PGC1 alpha is such an integral part of the FNDC5 pathway, and this FNDC5 eventually causes the release of irisin, and we know that exercise is playing a role in brain health, and if we knock out something in the pathway that's related to brain health, that you get lesions in your brain, then maybe this is the answer to why exercise is good overall for the brain. And again, if you overexpress and have a lot of irisin, one of the things that he was noticing is that BDNF levels also tended to go up. So here was that missing link. Exercise, healthy brains, increased levels of BDNF, increased neurogenesis, all borne out by a myokine as muscles started to exercise. So there's a lot of really big hope in this particular molecule. So one of the things that he did in his original paper in Ran et al. In 2013 was that he put mice on a 30-day endurance training. So they ran on a wheel, which is equivalent to running about 5K every single night. And one of the things that he found was that mice that were doing this, normal mice, not PGC1 alpha knockouts, but just normal mice, they did indeed increase the levels of FNDC5, not just in their muscle, which is where you'd expect it, but also FNDC5 levels within the hippocampus. And he found that this was responsible for their improved memory and learning, as well as neurogenesis. So the reason why he was really interested in this is that PGC1 alpha also seems to be important in this FNDC5 production. You can't have FNDC5 without PGC1 alpha. It is the step that is preceding the increased levels. So you have to somehow turn on PGC1 alpha, which then in turn leads to this increase in the FNDC5. And he thought this is an amazing discovery, because this actually accounts for why exercise is not just good for converting brown fat, which is what is happening here. Skeletal muscle, as it undergoes physical activity, as it starts to contract, will increase the levels of PGC1 alpha, activate PGC1 alpha. The skeletal muscle in turn will start producing more FNDC5, and more FNDC5 means that you can cleave more irisin, and irisin, as I've shown you before, can convert white adipose tissue into this brown adipose tissue that you see here, making the mitochondria much more energetically favorable to be able to use more insulin, to regulate body weight, etc. Irisin can also work directly on neurons, and it can cause them to increase proliferation, it can increase neurogenesis, a whole bunch of things. So here's this molecule that seems to answer all the questions that we needed to have addressed. How does this work, and why is PGC1 alpha in this particular pathway? So there are two different pathways, it turns out, ones that I'd like you to make sure that you know. Normally I don't ask you to memorize the entire pathway, but in this particular case, I think this would be important. This would be a muscle cell that you're looking at here. And this muscle cell that you're looking at, as it starts to contract, as it undergoes exercise, through the processes that you learned about in physiology, it will increase the intracellular levels of calcium. If you increase the intracellular levels of calcium, it will activate another enzyme known as calcineurin. It potentially could also activate calmodulin-dependent kinases, or CAMKs, which will then in turn activate CREB, that molecule that I told you about earlier, as well as some other pathways. Now, the one that I want you to concentrate on is CREB, or the cyclic response element, or cyclic AMP response element B. And why is that important? Activating CREB will cause more PGC1 alpha to be expressed in this muscle cell. And as you express more and more PGC1 alpha, it accumulates within the muscle cell. And again, eventually, we'll talk about the brain in a few moments. If nothing else occurs, if nothing else happens, if you've exercised for one day, because you know how you start off those New Year resolutions that I'm going to be like really physically fit, and you exercise for one day and you think that that's going to be enough to protect you from Alzheimer's disease, because I always fool myself into believing that, then actually gets broken down. It gets broken down by proteasomal degradation. However, if you keep doing it, if you keep undergoing exercise, we get this same pathway here, but we also introduce this other pathway. So we still get calcium entry as we did before, but instead of just activating this pathway, in addition to this pathway, we will also activate this pathway, which calcium will then activate AMP kinase, and AMP kinase will phosphorylate the PGC1 alpha. PGC1 alpha in turn can act as a transcriptional regulator to activate the transcription of FNDC5. Now, we've talked about FNDC5. As you exercise more and more, you produce more and more FNDC5 through PGC1 alpha. And FNDC5 in turn has that N-terminal domain that can act as a signaling molecule, which then gets cleaved, and this irisin molecule can then work on areas of the different areas like the brain, etc. Do you have any questions on this before I go on? I would like you to make sure that you know this signaling pathway, both signaling pathways, and the overall structure of the FNDC5 that I showed you earlier. So again, why is all of this so important and of big interest to us? We know that as you exercise more, that you are going to actually, in your muscle cell, your myocyte that you see here, the stabilized form of PGC1 alpha will drive the expression of FNDC5. It gets cleaved. This little orange portion would be the N-terminal protein sequence, and it will act on its receptor to convert white fat into brown fat. I've mentioned that on a previous slide. But more importantly, in the brain, if it gets its way into the brain, irisin will act on its receptor, the EF receptor, and it will actually turn on the production of BDNF. So the more you exercise, the more your muscles contract, the more irisin you form. Irisin goes up into your brain, interacts with different receptors, and will produce more BDNF. I just told you earlier on, BDNF is something your brain needs to be nice and healthy. So this explains and checks off all of those things that we were looking at before. Why do you get better or feel better after exercise if you're depressed? Maybe this might account for some of those different mechanisms. So this was a really amazing paper because it checked off all of those different things that people were kind of looking for. How does exercise impact your brain? Well, it might not be through GDF11, it might not be that blood-borne factor, but as your muscles contract, releasing irisin might be a viable alternative. And this was actually huge news. And this PGC1 alpha FNDC pathway, as it was published in Cell and in other journals, actually was thought to be really important. And we're going to come back to talk about different aspects of mind, body, and other factors later on. But we're going to see that this potentially was the answer to everything that people were looking for. And irisin was sort of the thing. Everyone wanted to synthesize irisin. People wanted to use it for depression. People wanted to use it to actually help for Alzheimer's disease, Parkinson's, any neurodegenerative condition. And this paper drew like a ton of interest, especially from the various drug companies. So do you like this story? Is this story any better than GDF11? Yeah, right? No. So irisin, this paper came out not too long after. So different drug companies like Novartis and other drug companies actually started to investigate, could we use this as a potential mechanism? So irisin, whenever you see a paper with this kind of title, and it's a myth, rather than an exercise-inducible myokine, this is actually not so good, right? It's like saying to you, like, actually, you don't really deserve a 4.0 in that course. You kind of deserve to fail that course instead. And it's kind of calling them out on whether or not this particular molecule that had made so much news actually was worthwhile. And again, you can read this yourself. But they went through and they just went to town on Bruce Spiegelman. So Bruce Spiegelman was under the same kind of criticism that Amy Wagers had. So remember Amy Wagers in GDF11? Here's this other paper coming out, not just by one, but by two different groups in Europe. So it was like a Europe versus America type of thing at the time, where all these people were coming out and saying, you know what, this whole PGC1 alpha, FNDC5, great story, but that's exactly what it is. It's a story. And what would you do if you were Bruce Spiegelman? Other than crying, yes.
I would ask them, like, what's your solution?
In science, we don't do, like, what's your solution to things.
I'm saying, like, it just sounds like a lot of people bashing on two scientists that try to find a solution and not really proposing a solution.
Okay, so I'm going to take you through Bruce Spiegelman's response, and you don't have to worry about this slide, nor do you have to worry about this slide. But let me tell you that the response by Bruce Spiegelman was very different than Amy Wagers. Amy Wagers, GDF11, it's been relatively silent. But Bruce Spiegelman, so just to put things in perspective, this came out his paper came out 2013, Ran et al. This paper came out in 2015. It was actually received in 2014, 1 year later. It came out early in 2015. This was sort of the gist of the paper. Everything that you've put in your paper, we can't really account for it. Your antibody sucks. The way that you do your research sucks. It's probably a really good story, but it's precisely that. It's just a story. This is Spiegelman's response. It's actually not a myth. And then he actually comes out later with this full paper. So this, again, think about the timeline, okay? So they received this paper in July of 2015. Do you remember when that last paper was published? It was February of 2015. So he's a really, really big researcher. So he basically had his entire lab working on this, submitting this as quickly as you can. Those of you who are in research will know that, publishing something within a few months is incredibly challenging, but he put his entire lab's assets to work to try to get past this story that iRisin is a myth. His whole academic career is riding on the fact that iRisin works, that it's real, and he has to be able to address all of these different issues that said iRisin is indeed a myth. So one of the biggest issues for us now in biology, and I've actually used this in my own slides, how do we detect proteins? What have I been showing you over and over again? So I told you about different markers. How did we see those markers? We stained them with antibodies, right? And the major gist of a lot of these different papers is your antibody sucks. So if you measure the protein directly, you would actually go around that whole problem. You're not using an antibody. And so Spiegelman decided, I'm not going to measure it using an antibody. I'm going to go directly after identifying and showing you the protein exists for real, like within the blood. And again, this is a great example of a visual summary that all of you are busy working on and making beautiful, right? Ready for next Wednesday? So I'm around, by the way, on Friday, so if you want to drop in and go over your visual summary and get some feedback, I'm happy to do so. So let me tell you a little bit about Spiegelman's response, and I'll try to wrap things up in the next 5 minutes or so. These are things I want you to make sure that you know. Why did people have a problem? Why did Novartis, this huge drug company out of Switzerland, have this problem with his research, etc.? So these are all of the reasons why, and I'd like you to make sure that you know this slide. So do you know what ATG normally encodes for? Those of you who are in biochemistry. Biochem 210, ATG? Sorry? Methionine. So it's your start sequence, right? So you always encode a methionine, and so ATG is sort of the canonical start sequence. If you look at proteins, proteins always start with a methionine, and we always have an ATG. iRisin does not have that. iRisin at the N terminal actually has an ATA. It's a non-canonical start sequence. So people were looking at that going, wow, this can't possibly be real, can't be a real protein. It doesn't have an ATG start codon. So ATA being there made a lot of people suspicious. People thought it can't actually be a real gene either. When they looked at the structure of the gene, they said, yeah, you know what? When you look at the gene, it probably doesn't form a real protein. It's probably just a pseudogene. It's not transcribed. It's not translated. There's a sequence for it, but actually it probably doesn't exist directly. What you're picking up in your papers is an artifact of your antibody. So this is that issue with the antibody-based detection. When they went in, they actually couldn't detect any iRisin in their samples. So that was a big issue for them. We can't detect it. How could you possibly detect iRisin since we can't, following your own protocols? It was not reproducible. People just couldn't get the same results as Bruce Spiegelman's lab did. So he decided to get around all of these different issues, and he decided to use tandem mass spectroscopy. You can actually take a blood sample, fractionate it using spectroscopy, and actually see if your protein exists directly in the blood. No antibodies, nothing else. So he decided, I'm not going to bother addressing all of their issues. I'm going to just show them that the protein really does exist. I can't address this ATA. Other molecules have ATA. I can't address the pseudogene, but I can show them that mass spectroscopy can pick this up at a physiological level. He also wanted to show that exercise does indeed, using this method, elevate the levels of iRisin just as he would have expected. The only thing that he did, which is a little unorthodox, is he used a deglycosylated sample, but he also addresses this in his paper as well. Like, I need to get the purest, best source of protein. I need to reduce all of these contaminants like IgGs and albumin. So I'm going to deglycosylate. I'm going to purify my sample before doing mass spec. Again, this is sort of his overall setup. He would have a sedentary group versus an exercising group, analyze their blood samples using tandem mass spec, and then show very, very clearly that he can actually detect irisin in their blood, and that exercise actually elevates irisin levels even higher. So again, you don't need to memorize all of the details on this slide, especially the stuff over here on the left, which shows you the detailed way in which he took samples out of a gel and then ran it on mass spec. But do you know any of these molecules? So I mentioned to you, for example, adiponectin, which is up here. And adiponectin is normally found in the blood. He's able to detect that on mass spec. He's also able to detect leptin. Have you heard of leptin before? So he detects leptin in the blood using mass spec, and here's irisin. And so he's actually making a point that we are using tandem mass spec. This is in the physiological range of other molecules like leptin, like insulin. So insulin is down here. If you guys can't pick it up, that's not our problem. That's on you. So he's going after them very succinctly, and he actually is able to show that in his blood sample, not only does he have all of these other naturally occurring, physiologically relevant molecules, but he also is able to detect irisin smack dab in the middle. So it's at a physiological range, somewhere between leptin and insulin. So people should be able to detect this particular molecule. He's not using an antibody. He's detecting it using mass spec directly. So the only way he could detect it is if the protein actually existed. And then his response is actually really good. If you have a chance, because it's only about 3 pages long, you should actually read this paper. It's like such a fantastic paper. You never get to see a scientific paper that actually has this type of language in it. So he actually goes directly after them. You said that this whole thing was a myth, so he's really pissed off. So he starts off with this. Whenever you see that, you know, we made the claim or you claimed something, again, it's never a really good thing. He also goes directly after them, where he's developed a quantitative, precise, unbiased assay for detecting human irisin. This assay definitively shows, when you use words like definitively, it's saying don't ever question us again. This is like for all time. Don't ever question that we've been able to detect this. And again, we also confirm our earlier report, so kind of going back to take a little dig at the different individuals. So this is how science kind of responds, right? So no one's been able to go back and refute Bruce Spiegelman since then. People kind of are thinking that irisin might be the actual molecule that improves mental health, your brain function, everything else after exercise. Again, you can read through the rest of his sort of response to the criticism that he received. Do you believe that irisin exists? Yes, big time? Until the next paper comes out that says that it doesn't, right? Because that's, again, you can also be assured that the group in Novartis is probably also working on a rebuttal to his rebuttal as well. Do you have any other questions before we wrap things up? So again, for those of you who came in a little bit later, I will be posting your adjusted individual marks if they do indeed go up based on your group. But please make sure that you check your unadjusted marks first over the next couple of days. If you need to come in and see me about your visual summary and get some feedback, please do so on Friday or Monday of next week. I'm hoping that you're all taking care of yourselves. Have a great weekend. I'll see you next Wednesday.